Hey everybody. Yeah. So here we're going to start with a very crucially important chapter, right? In botany. Just hold on a sec. So I have to admit one thing. Just hold on. Right. So this workshop is going to be on embryology. Has anybody heard of this field, embryology? So let me tell you, this is about studying embryogenesis, or in other words, developmental aspect of a plant. In more usual terms, an angiosperm. We usually study the development of an angiosperm and then we move ahead with, because this is one of the most advanced kind of development that we see. One more thing, when we discuss about any kind of development, right? There is a certain pattern that we follow. Usually, most of the professors, when they teach a particular embryological concept, be it botany or zoology, the flow would be gametogenesis. When I say gametogenesis, it basically means formation of spores, right? The male microspore and the female megaspore in case of plants. So we first study microsporogenesis and megasporogenesis. So we separately studying the male part, the female part, the formation of gametes. Now once the gametes are formed, we talk about the route that it takes to fertilization. So we don't directly jump to fertilization. In case of plants, we talk about the passage or the route for the gametes to fertilize, which is nothing but pollination, right? In case of angiosperms. So we have a very detailed discussion on what is pollination, what are the kinds of pollination, right? There is autogamy, geitonogamy, xenogamy and all that, and the modes of pollination, various pollinating agents and all that stuff, right? Now once we are done with pollination, we discuss about what happens post pollination, and that is where we're going to discuss about fertilization. Now, once we are done with fertilization, we talk about post-fertilization changes. So everything before fertilization, it was that is the gametogenesis part. That was called, that is called the pre-fertilization events. Let me write this down. There. So I'll come to that. Before that, let me give you a very brief introduction, whatever I was saying so far. So as I was saying, there is pre-fertilization events. Followed by that, there is fertilization, and followed by that, there is post events, right? And in pre-fertilization, we discuss about gametogenesis. Here we discuss about pollination, and here we discuss about development. Right? We discuss about all that. PEM, right? Endospore, and all that. So this is going to be a flow. In fact, for every single embryological chapter of any plant, we have to study the pre-fertilization events, the process of fertilization, and the post-fertilization changes that happens in the plant body. Another thing, before that, let me also introduce myself, right? Since this is a workshop, I'm sure most of you don't know me. I'm Ritvik Surya. I'm doing my BSc, currently pursuing in life sciences, which includes botany as well as biotechnology and various other fields. Along with that, I'm also doing research on chemistry, because technically that's a part of life science, from Christ University. You might have heard of it in Bangalore. And our first lecture is going to be on the very first aspect that we were talking about right over here, right? Gametogenesis. Now, gametogenesis again, um, it has microsporogenesis and megasporogenesis. What is microspore? You know what is microspore? That's the male gamete, right? And in case of plants, then what is microspore? This is pollen. Pollen is the male gamete, and over here this is embryo sac, the female gamete. So we're going to study basically the development of this. More than embryo sac, I would rather say that embryo sac which contains the egg along with the egg apparatus. We'll discuss all this in a lot of depth. Right? And one more thing I'll admit straight away, the presentation which I prepared for you guys, I was given this whole lecture notification in a very short span of time. I wasn't able to prepare a very good presentation. Usually my presentation has got only pictures, right? And I speak out and explain words and I make notes like I'm doing here. But for this particular lecture, I had no clue until a couple of days back, and all I could do was take screenshots from my handouts, my previous lecture handouts, and as it is paste it here in the presentation. So I'm really sorry for that. I'll try my best. What I'll be doing is I won't be reading it as it is. I will be explaining you the concepts, and you will have the notes on the screen. Now, I thought there's one advantage to this. We can move a lot faster because now you don't have to take notes. I don't have to prepare special handouts for you people. Everything is in the presentation. Like you can see, most of them are like text, right? So the entire presentation has got text. So I won't be reading it out. I would be explaining it to you, but you will have the text with you, which means you don't have to really concentrate on taking the notes and all the other factors. You can free up yourself from that burden. Okay, so let us get started, right? Some very basic concepts on the structure of flower. Now when I say flower, the first thing that should come to your mind, rather, it is flower. Yeah, that word strikes a note. We're talking about one of the most advanced reproductive structure. So a little note on evolutionary concept, right? So you might have heard about this as well. So there were a lot of spores initially. So you have zoospores, right? Oospores and chlamydospores. Then there was conidia. So all these are asexually reproducing structures. They tend to support asexual reproduction. And this was prevalent mostly in algae. Okay, we have discussed this in another workshop, biodiversity. In that workshop, I've explained everything in a lot of depth. It was probably was 5 to 6 months back. We'll be conducting that workshop again this summer. I'll be again conducting the same workshop. Anyway, we can have a look at that. So those were the most primitive reproductive structures. Post that, if you're talking about pteridophytes and gymnosperms, then we talk about Microsporophylls. Very interesting structures, right? Microsporophylls, and they were present in structures called conidia. Sorry, not conidia, cones. They are present in structures called cones. Cones is also known as strobilus. So strobilus were these structures, these tiny little, structures containing various leaflets. Structures like this. And it contained these leaflets. These leaflets were called microsporophylls. So basically, if you take a longitudinal section of this, you could see that it was something like this, right? Something like this. So you could actually, I'm drawing it very small now. So you might have seen this diagram when you were studying gymnosperms and all that, right? So it contained, structures like these were called microsporophylls or megasporophylls. Usually these were microsporophylls, and the one at the bottom, which was more spherical, this was the megasporophyll. And they contained something called as microspores and megaspores. Right. So this was an evolutionary upgrade from spores, from asexual spores to sexual spores. There are various kinds of spores, right? Spores can be sexual as well as asexual. Chlamydospore, for instance, is an asexual spore. Zoospore is a sexual spore. So various kinds of spores, and then from there it comes to strobilus and microsporophylls and megasporophylls containing microspores and megaspores. Now these become more advanced in angiosperms. Now angiosperms, or what we call as flowering plants, are the most advanced. So much so advanced that they had a altogether different structure for reproduction called the flower. Flower is nothing but a kind of reproductive structure. Okay. Now in this reproductive structure, you have various groups of layers, right? Layers, or what we also call as various strands. Now in case of usual, like, there are a lot of exceptions. You can have flowers which are bisexual as well as monosexual or unisexual, but in a usual bisexual flower we see that there is a gynoecium. Now this is this entire structure over here. This is the gynoecium. This entire structure is the gynoecium. Okay. Now gynoecium is made up of carpels. Clear? Gynoecium is made up of carpels. This is what it says, right? Gynoecium members are called carpels. A lot of carpels together, because you can have unicarpellary, bicarpellary, tricarpellary. There can be multiple units of carpels which make up a gynoecium. This is a unicarpellary gynoecium. Then there is the male structure. This is the male structure. And this entire male structure is called androecium. And this is made up of stamens. Stamens. So you can have again multiple stamens. You can see that now there are actually there are 4 stamens over here. Right? 1, 2, 3, and 4. Right? 4 stamens. It can be a definite possibility. So that is why we say that androecium, the members are called stamens. Okay. Now after that, there are other non-reproductive structures which don't. Now this you might also know, you should know rather, that this is the male gametophyte and this is the female gametophyte. Now for the rest of the lecture, I may not be writing the word male and female. You should know that this is representation for male and this is representation for female. Right? And for fertilization, what is the representation? Here. This means it is fertilization. Okay. Anyway, so this is the reproductive structures. Androecium and gynoecium made up of stamens and carpels. Whereas there are other structures which are actually important for the process of pollination or just as a part of it. One such is sepals. Now these are called sepals, and the whorl of sepal, right, containing multiple sepals. This is a sepal and its whorl is called calyx. Similarly, this is a petal. This is a petal and the whorl is called corolla. So you should know this, right? Corolla has multiple petals. Calyx has multiple sepals, androecium has multiple stamens, and gynoecium has multiple carpels. Remember it that way. Okay? So these are the four important whorls. Now we call this the whorls. We call this the whorls of a flower. Is everything clear in this slide? I'll be going slide by slide and explaining every concept very slowly. Okay. Now, yeah, one tiny thing which I missed out here, that very simple stuff all these are. You should, or you rather, I am pretty sure you do know that carpel, if you're talking about, it's made up of stigma, style, and ovary. This part over here, this is the stigma. This line-like thing over here, that is the style, and this whole bulgy thing is the ovary. I'll explain each one of them in much detail when you're studying megasporogenesis. Similarly, if you're talking about androecium, then this part over here, this is the anther made up of, this is known as the anther, and this whole line over here, this is known as the filament. Clear? So I can rather say that androecium itself here, this is made up of anther and filament, or rather stamens, right? Stamens is made up of anther and filament. Similarly, carpel is made up of what? We just discussed here, right? Carpel is made up of stigma, style, and ovary. These three things make up the carpel. Clear? So these are important stuff, okay, and these are very basics as well. I'm pretty sure you all know this. Trusting on you for that. Moving ahead, right? Moving ahead. We are going to start with microsporophylls. Basically, we're going to start with microsporogenesis. This lecture has got microsporogenesis components in it. I'm pretty sure it will take multiple lectures to complete microsporogenesis as such. If you're talking about microsporogenesis, I said this is made up of microsporophylls or stamens, and we also know that stamen is itself made up of anther plus, what is the other thing I said? Filament. And that is very evident in this diagram as well, right? Here you have an anther. This whole thing is an anther. And this whole thing is a filament. Now, interesting thing, we'll go with a little ultrafine structure of anther. Then we realize that anther has got two lobes. It has got two lobes. This is, I'm talking about the usual case. It can be trilobe, tetralobe as well. We will discuss more exceptions. But usually, anther, if you're talking about anther, right? And I say anther, it is only this structure, right? It is bilobed. Right. Two lobes are present. And between the two lobes, you have a tissue. That tissue is called connective. So I can say that bilobed, attached by connective. Clear? Now, if you're talking about a single lobe, right? Take a knife, and this is, if this is your anther, right? Bilobe structure with the connective. Take a knife and cut through this plane, right? Cut it. So I'm placing a plane, I'm passing a plane through this region. So what do I see? So as a result of this, I see this kind of a structure basically. So this is one lobe and this is two lobes. Two lobes are present. Each lobe has got two chambers. And this is known as a pollen chamber. pollen chamber. So I can say that each lobe has got 2 pollen chambers. Clear? Now these pollen chambers has got tiny little pollens in it. If you're talking about this again, pollen chamber, lobe, pollen chambers, right? And here you have the connective. Now inside this pollen chamber, you see tiny little, granular substances. We call this granular substance. Now these are called pollens, and this is nothing but the Microsporophyll or the male. This is not exactly a male gamete, because male gamete is something formed inside the pollen, but this is a very brief general overview of the Microsporophyll. These are Microsporophylls or pollens inside the thing, and that is what is explained in these lines in here. Okay, and even this line over here, right? It explains the same thing. Any doubt in this slide? As such, we haven't discussed about filament. In fact, we are not going to discuss about filament. This is a vegetative structure. Always remember, when you are studying embryology, we give importance to certain structures, right? Now, structure can be either vegetative or reproductive. Now, when I say vegetative and reproductive, again, vegetative means we take it for granted that this is diploid, okay, and we call this sporophytic. And if you talk about reproductive structures, usually this is haploid, whereas we also see that there are some exceptions. There are some diploid, reproductive structures, but none of the vegetative structures are haploid. But most of the reproductive structures are haploid, and we call them as gametophytic. Because they bear gametes, probably that's why we call them gametophytic. Now, we are always interested in gametophytic structures. We are not interested in sporophytic structures whatsoever, because these, the genetic material of the sporophytic structure does not take part in fertilization. No fertilization for them, which means this genetic makeup itself is not utilized anywhere. It is not expressed anywhere. So we are not bothered to study that. We only want to always remember, right? Sporophytes, they are supportive structures. They support gametophyte. Remember? Okay. So that is what is again explained in these points. You can study that. You have the notes with you anyway. Moving on. Now let us study about the development of anther. I'm going really slow. Let me know if I can go faster, because that will explain, and I'll be able to cover more concepts in the limited amount of time that we have. Okay? Yeah. Anytime you feel that it's getting, very childish or very easy, remind me to go a little faster. Now things get a little more complicated. We're going to talk about the development of anther. Again, reminding you, this is what we are interested in, anther and not the filament. How is this particular structure developed? Now, this is not a single cell. It's a lot of cells. It's a macroscopic structure. So it has got its own tissues, right? It's got its own tissues. Now that is what we are more interested in. If you have a look, each lobe, read these sentences. They are very clearly explained, right? They have got a hypodermal region. This is basically the hypodermal region. This thing. Now you all should know what is epidermis, what is hypodermis. Now the outermost is, obviously we have some, if you're talking about sections, we obviously have cuticles, but since this is not a section of a vegetative structure, we don't have a cuticle. We have an epidermis. Then we have an hypodermis, the inner structures. After that, if at all present, there would be an endodermis. After that, if present, there can be a cortex. After that, if present, there can be a pith. This is something you study a lot more detail in anatomy, which is the next workshop which I'll be taking. So if you're talking about hypodermis region in here, now these cells over here, right? So these cells make up the hypodermis. Probably the second layer. They are the hypodermis cells. And what tissue is this? These are basic parenchymatous tissues. Okay, they are all parenchymatous. Okay, hypodermis. Now what happens to hypodermis? This is of concern, right? Hypodermis start dividing. Okay, some of the cells, not the whole of the hypodermis, some of the cells start dividing. Now these are the cells which are large and which have radial growth and which have dense cytoplasm and conspicuous nucleus. Now understand each of these terms. The hypodermal cells, okay, the hypodermal cells, it must be hypodermal, it must be a large cell, and it must have a radial growth. What do you mean by a radial growth? It should be a little elongated. Okay, radial growth, and it should have a dense cytoplasm. What do you mean by dense cytoplasm? This is a kind of cell we are talking about. Cytoplasm should be dense, and the cell wall must be smaller and smaller. Now again, here various terminologies which you might be very familiar with. If you're talking about a cell has got a cell wall under which there is a lumen, right? Now this is a lumen, and this is the wall. Now if the wall gets larger, the lumen gets smaller, and that is what we call as a sclerenchymatous cells. So if you're talking about a sclerenchymatous cell, it will be something like this. Huge wall, very narrow lumen, so narrow that the cell cannot live any longer. It can't perform any metabolic functions. Whereas if you're talking about usual parenchymatous cells, they have got very large lumen. Why? Because the wall is extremely narrow. Huge lumen, very narrow wall. So we want such cells, which has got a very huge, dense cytoplasm. Dense cytoplasm. Criteria mixed. And after that, we need a conspicuous nucleus. What do you mean by a conspicuous nucleus? The nucleus is supposed to be large, and it should be very well defined. It should be conspicuous. So such cells, right? These are the cells which are known as archisporial cells. Guys, very important term. This is also known as microspore, or rather let me change that word, because if I call microspore, then you might get a little confused. Let me make this microsporangia. Microsporangia initials. Clear? Archisporial cells or microsporangial initials. Now what are these cells? These are the large conspicuous nucleus dense cytoplasm cells found in the hypodermis of the tissue. Okay, anther tissue. Now these cells, what will they do? They will start undergoing periclinal division. Okay, what do you mean by periclinal division? Again, simple terminologies you should know. This kind of a division, right? Division at the bottom surface, right, forming two lateral cells. Now this is known as a anticlinal division. Anticlinal division. And a longitudinal section forms a periclinal division. Clear? So what will these cells do now? They will undergo periclinal division, which means what? Initially I might have had one archisporial cell here. Now this will divide, right? This will divide and form two archisporial cells. This will again divide and form three archisporial cells. Again divide and form four archisporial cells. Now gradually what is going to happen over here is, if this was my sporogenous, if this was my anther tissue, it's going to form a complete layer of these, archisporial cells. There's going to be an entire layer of archisporial cells. Are you getting this? Now these archisporial cells post division, right? It consists of 2 to 3 cell wide plate running along entire length of the lobe, which means this entire length of the lobe will have a surrounding boundary kind of cells, all of which are archisporial cells. Okay. Now what will happen to these archisporial cells? They will start undergoing anticlinal division once the entire wall is surrounded. Once you have this kind of a structure, now they will undergo anticlinal division. So basically now what you're going to get here as an end product, you will have obviously one row of archisporial cells. You will have another row of archisporial cells, right? There is one row of archisporial cells and another row of archisporial cells. Now the first row over here, right? Understand this very clearly. First underwent periclinal division, formed an entire layer. Now they underwent anticlinal division. This was periclinal, right? So we had to select this. Periclinal division, again periclinal division, formed the entire surface boundary. Now this boundary cells, right, they start undergoing anticlinal divisions. They start opening up like this, right? And they form what? What is the end result? We have two layers, right? Now one of those layers is known as primary parietal layer, and another of that layer, right, the inner layer rather, is known as primary sporogenous layer. Clear? So now basically I have the anther wall with two layers. One of them is the parietal layer, the other is a sporogenous layer. Has anybody got any doubt in this concept? And that is actually what your entire line explains, or the whole text, right? I'll send you 2 PPTs, okay, after the end of this entire workshop. One PPT with all my notes embedded in it, okay, and another PPT which is completely blank. Okay, so I'll be sending that. Do remind me to send you the same. Okay, sure. Oh my gosh, for that I haven't made a copy of a blank PPT. Is that okay if I send you only the notes PPT? You can erase it whenever you want. I'll be sending you an editable copy. You can erase all the footnotes, whatever I've written here, if you want to read something. Okay. Otherwise as well, I'm pretty sure the text is visible. Okay. So that is what is explained in this region. That one paragraph in there. So now at the end of the story, right, what do I have at the end, right? This is what I have. Okay, not very clearly explained in this diagram. I'll come to this diagram later. For now, I need to understand it from the previous slide. All I have is, I have obviously the anther wall, and then there are 2 layers. The first layer is the parietal layer, and the second layer is the sporogenous layer. Okay, now what happens next, right? This parietal layer gives rise to the anther wall structure. Parietal layer in the sense the outer layer, right? So if I had this kind of a structure, the outer layer gives rise to anther wall. But wait a second, there is a problem. Anther wall is made up of multiple layers. In fact, it's made up of 4 kinds of layers. So what is going to happen now? Since I have a single layer of parietal layer, which has to form 4 layers of anther walls, obviously I need more. What kind of division do I need more? Periclinal or anticlinal? Anticlinal division. So now I need 3 more anticlinal division. And so that happens. Are you getting this very clearly now? Right? What is going to happen? So initially I had a single layer of archisporial cells. All of these were archisporial cells. Right? Let me write it here. I had a single layer of archisporial cells which differentiated into a parietal layer and a sporogenous layer. The parietal layer forms the anther wall, and in order to do so, it differentiates into 4 kinds of layers, which is the epidermis, endothecium, middle layer, middle layer is, it can be single or multiple middle layers, and then there is tapetum. Now these are the 4 layers formed by which one? The parietal layer. Now parietal layer does not exist anymore. It has completely converted and formed epidermis, endothecium, middle layer and tapetum. Now the parietal layer has got no existence. Right? But we still have a sporogenous layer. So basically now what is the anatomy? If you see very carefully, you have the epidermis, then there is endothecium, then there is middle layer, then there is tapetum, then there is sporogenous layer. And then obviously there is the rest of the cortex. This is the anatomy. Right. This is the anatomy so far. Now what will happen next? Obviously we have an unknown layer there, right? Sporogenous layer. I never spoke about sporogenous layer. That is because sporogenous layer again undergoes multiple divisions, and that is what forms the microspore mother cell. Okay, so eventually this thing will start getting degenerated and it will form the microspore mother cell. I'll come to this within the next 1 or 2 slides, but until here is everything clear. I'll explain you what are these 4 layers in detail in the next slide. But did you understand the division? So first we had the periclinal division forming more and more archisporial cells. Then we had the first anticlinal division forming a parietal and a sporogenous layer. Then we had more anticlinal division which divided the parietal layer into 4 kinds of layers which together form the anther wall. Is this clear? Does anybody have any doubt in this, guys? Please tell me. Okay, I'll brief you out one last time. Listen very carefully. Let us talk about a single cell. Let us not worry about this huge number of cells that we are talking about, right? Forget that there is a tissue. So I have a single cell, okay, a single normal cell. Now what are the characteristics of these cells? This cell has got a huge dense cytoplasm. It has got a very narrow lumen. It's got a very conspicuous nucleus, and the cell is large, right? And the cell is radial. Now these were the characters I listed out in the previous slide. Now such a cell, a single cell, right, with all these characters, now it will undergo a periclinal division and it will form 2 cells. It will further undergo more periclinal division to form more cells, and so on. Now this is nothing but the archisporial cells. Now these archisporial cells will now undergo anticlinal division to form 2 layers of this whole structure. Now these 2 differentiated layers are nothing but, one of them is parietal layer and another layer is sporogenous layer. Now the parietal layer will further undergo division to form 4 layers of cells. not exactly 4 layers, 4 or more layers of cell, plus the sporogenous layer. This is the sporogenous layer, it is as it is. Now these 4 layers together form the anther wall. And the 4 layers that I was talking about, you can see over here, right? We have epidermis, endothelium, middle layer, tapetum. That is the order in which it flows. And inside that, ultimately, don't worry about this thing at the moment. This is something formed by the sporogenous layer, right? So up to here is this clear. There are just 4 layers, and then there are some more sporogenous layers. This forms a sporogenous tissue. Sporogenous tissue will further form a microspore mother cell, as I said, which will further form the microsporangia, and those microsporangia would be residing in this space. But that is not what we are bothered about. That will form the pollen. Microsporangia will form the pollen. But we aren't worried about that at the moment because we haven't touched the topic. What we are worried about is the outer cells, the parietal cells which forms the wall. Now let us discuss about these 4 layers in detail. Yeah. So the first layer was epidermis. Now what are the characters of epidermis, guys? This is something which I won't be discussing in too much detail. You all are very good at this. Now, epidermis has got the outermost layer. Right. This is obviously epi means what? Epi means out. Epi means out. This is the outermost layer. It undergoes only anticlinal division. Obviously, it has to undergo only anticlinal division. Yeah. And cells become greatly stretched and flattened in order to keep pace with the enlargement of anther. This is one thing you should understand. Anther is actually growing because there are a lot of cell division. A lot of cell division will in turn increase the girth of the tissue, right? It will increase the thickness or the size of the tissue as such. Which means what? As the anther starts stretching and growing, eventually these cells will become stretched. They become almost flattened. The initially almost spherical cells would have now become really elongated and elliptical. Is this logically correct, right? That is what has to happen. Now eventually what will happen in dry habitats? Now this is in regular plants. In case of desert plants and in case of dry habitats, they lose contact. These epidermal cells lose contact. Obviously when I stretch this even more, right? When I stretch these even more, what will happen? They will lose contact because they can no longer hold each other. Yeah. And they become the withering remains. So at maturity, you don't see a clear epidermis. Epidermis is not continuous. At maturity, what happens? At maturity, you are basically able to see only some withering remnants or some leftover of the epidermal cells. Why? Because it is stretched so much. From such a tiny size, it would have stretched to such a huge size. So obviously the epidermis is withered. Is this story clear about epidermis? Moving on. Endothelium. Now in case of endothelium, what do we see? Beneath the, endothelium is obviously, first thing, endothelium is beneath the epidermis. Right below the epidermis. And as such, yeah, again, if you can see the notes, right, it's very clearly explained here. So basically it is also getting elongated, right? It undergoes a very similar process. So this is the same fate with endothelium. It gets more and more elongated. Ultimately the cells start becoming detached from each other. Clear? Now I don't know, should I explain this to you or not? Something about endothelium is very important. It is actually mentioned in the last line. The opening through which the pollen grains are discharged from the pollen sac is called stomium. But I'm again worried. Should I explain this to you or not? Because I haven't discussed pollen grains as such. Remember one thing, endothelium is an opening. So basically if I have an epidermis, endothelium would have a lot of openings. And obviously, as I said, as I haven't explained yet, but I have just given you a clue that sporogenous layer will ultimately form pollens, which will be inside now. Now these pollens, don't worry about middle and tapetal layers, they are very weak layers. Okay, these pollens find endothecium as points to exit, points to dehisce, and that is something actually happening. It happens. That's what is mentioned here. And these openings, tiny openings where the dehiscence happens, this is known as stomium. Clear? If not clear, forget it for the moment. We'll be discussing this again. Now, nothing much to say about middle layer. They don't play a very important role in reproduction as such. But middle layers have a very important role when it comes to various exceptions and various taxonomic characteristics of a plant. If you found a new plant, right, and you need to taxonomically classify it into one of the classes, then you should know some of the unique characters, and middle layer becomes one of the most unique characters. Why? Because middle layers, okay, first of all, general characteristics, okay, they're at the middle, so they're called middle layers. Now they can be 1 to 3 middle layers. That's why I said it is layer, or it can be a single middle layer. It could be number of middle layers. There are a lot of exceptions. So here again, with examples, right? There are 3 to 4 layers when you talk about holopetalia. There is 2 layers when you talk about ranunculus, and there are several. Several means more than 4, greater than 4 to 5, right? Several layers of middle layer when you talk about lilium, right? Whereas if you talk about wolffia, right? Wolffia, the middle layer is absent. So are you getting the various kinds of exceptions that we see here? So again, remember the this content actually you should remember with examples only. You shouldn't remember it just like that. So holopetalia, it has got what? How many layers? It has got 3 to 4 layers. Now if you're talking about ranunculus, it has got how many? 2 layers. If you talk about lilium, it has got more than 5 layers. If you're talking about wolffia, it has got 0 layers, absolutely 0 layers. Clear? These are very variable kind of cells, and they don't play a very important role as well. Yes, the cells are flattened, the cells are crushed. And next time, the meiotic division in the microsporophylls mother cell. Yeah, whatever general characteristics of the cell, they undergo meiotic division as such. They are flattened, crushed, and variable numbers. With examples, you should remember. Holopetalia 3 to 4, ranunculus 2 cells, lilium 4 to 5 or much greater than that, wolffia no middle layer. Now we'll come to the most important layer, students, that is tapetum. Now, tapetum is a very important layer because this is something which helps in nourishment. This helps in, very typically it helps in nourishment, but obviously there are other roles of tapetum as well. New research is showing much more new roles of tapetum as such. I shouldn't have compressed everything in a single slide. I'm finding it very difficult to explain here. I'm not able to write anything because whatever I write is wiping off the content. Yeah, my bad. I'll see to it that it doesn't happen the next time. Let me explain here itself. There are 2 types: glandular or secretory tapetum and amoeboid tapetum. 2 types. Clear? Okay. Now understand this. Cells remain in situ. Cells remain in situ. Then it is called glandular or secretory tapetum. And if the cells break down but the protoplast remains intact, right? Then it is called amoeboid, which means what? Consider tapetum as a layer. As such, it is a layer, obviously, where if I have a cell like this, right, if this is a anther wall, and these are the tapetum, right? Now if tapetum forms this kind of a structure, it is all connected, then I call this what? Glandular. Whereas the cells are broken, and if it is inside, then we call this amoeboid. Basically anything that has got indefinite consistency or indefinite shape or anything indefinite, then we call this amoeboid. That is a usual term. Don't confuse this with the protoplasmic motion, anything of amoeba. It's not like that. It is just that it is indefinite. It is just roaming around inside the nucleus. That's why it's amoeboid. Okay. Now there is a reason why we call it glandular, because glandular helps in secretion. That's why it's also known as secretory. Okay, it helps in secretion. Okay. Now come to this topic over here. Towards the close of the meiotic division, no, not important, not important, not important. Yes, here. So tapetum is active, especially glandular tapetum. It's active in synthesis of all major class of compounds such as proteins, lipids, carbohydrates, etc. And the levels of synthesis are state specific. Now, what does this paragraph basically try to explain you? These are functions of a glandular tapetum. What do they secrete? They secrete such macromolecules. Okay. And what does the meiotic tapetum do? As such, it has got no much role. Yes, definitely it does help in nourishment. Right. Okay, that is one important role of amoeboid. Now, also one more thing, when you're talking about glandular tapetum, it helps in the formation of sporopollenin of the pollen walls. Now, this is important because we will have a question next. What is sporopollenin? So, always remember sporopollenin is a very important compound in the enzyme. What is an enzyme, sir? I'll explain that to you in the next slide. Remember, it's a very important compound in the enzyme. Enzyme is made up of rather sporopollenin, and this is one of the most unbreakable, or I can say undecomposable substance. Now, even enzymes can't break this. They cannot take it. Okay, and sporopollenin is very important. It's important for protection, various stuff. You can't make a hole in sporopollenin. Basically, it's one of the most strongest polymer in plants. Okay. So are these four layers very clear to everyone? Epidermis, endothecium, middle layer, and tapetum. We've discussed all the four layers. Do read the notes, whatever it is in this paragraph. Now you also see large vacuoles in cytoplasm. Yes, nuclei begins to show signs of degeneration and all that. Towards the close of meiotic division, yeah, this is what I was saying. Tapetal cell becomes close contact with each other. Now, all of these layers, in fact, have the same kind of consistency or same kind of thingy, right? As I was explaining here. So again, even if you're talking about tapetum, initially tapetum would have been like this, thickly packed. Towards the end, it will become like this, very far away from each other. Why? Because the whole thing is stretching. It is like a balloon. It's stretching. More and more cells are formed at the center, which means the outer cells with a limited cell number, initially they try to elongate, they try to stretch as much as possible. Ultimately, when that limit crosses, the cell becomes separated and you have very degenerated set of cells. Now that is how most of these degenerate, and that is actually very much logical, because as the plant is maturing, there is no need of tapetum. As the plant is maturing, there is no need of, say, middle layer, or there is no need of epidermis. So as maturation progresses, whatever is not needed has to degenerate. After fertilization, the synergids has to degenerate, right? Now this degeneration has to happen, and that is taken care with the help of growth. Growth being a very important factor, because growth will cut down everything from the sides. Is this clear? As it starts growing, it starts breaking down and cells start getting detached from each other, and eventually you'll have nothing. Okay, Baba, then we'll start with sporogenous layer, shall we? Or you don't want sporogenous layer for the day? It's a little big topic. We'll do one thing, we'll keep sporogenous layer and the rest of the content. Oh, there's so much. We'll keep all this in the next lecture. Okay? I was planning to complete microsporogenesis today, but looks like I can't even start microsporogenesis. So I'll go back to all the slides we discussed about. Any doubt in this slide? I'm pretty sure, I hope not. Any doubt in this slide? Don't hesitate, you can ask me. In this slide? Sorry, this slide? This slide. These are the 4 slides we did today. Okay, then sporogenous layer onwards we'll discuss in the next class, because sporogenous layer is a very important layer. That is what helps in the formation of microspores. It's very important and essential in microsporogenesis. If you understand sporogenous layer very thoroughly, then that is the end of the difficult part. After that, morphology of pollen structure, I won't even be explaining all this. You should understand this by yourself. I have not even bothered to explain that. Pollen division, little bit I'll explain. It's a little new topic for you. Pollen allergy, pollen storage, pollen viability. No, I won't be explaining all this to you. Those are very easy chapters. Yes. Chalo, it was a very good interaction from your end. Yeah, almost 1 hour now. Closing the class a little bit early, today being the first lecture. In the next lecture, we'll try to cope that up. You want me to mention the next lecture? Next lecture will be on, let me give you a title slide here. The next lecture is going to be, rather, the first lecture we never discussed microsporogenesis, right? So let me change the topic. Let me write it as, what did we discuss today? I can write this as ANTHER wall. That was the first lecture. Okay, that was the main content we did, right, for most of the time. In the next lecture we'll do microsporogenesis. This will be our lecture 2. Okay, so do remind me, do study all this. I won't be able to share these slides with you right now, because if I share this, along with this we'll get all the other slides also, and that will be a little confusing. Some of you might overread, some of you might not read, some of you might get confused. So in order to avoid all such mess, I'll not be sharing it. I'll be sharing it at the end of the seminar. Okay. Chalo then. See you everybody. Meet you in the next class. Good night. See you.